Okay, sorry about that. A little bit of technical difficulties. We'll go ahead and begin. So today we're going to finish up our discussion on the relationship between sequence and structure and function, and then we'll also talk about how you make peptides and proteins chemically in the lab. When we were last together, we had examined this amino acid sequence, and we had pointed out that at regular intervals in this sequence, you see nonpolar residues, with some exceptions, but in general, they repeat every 3 and then every 4, almost as though you have this repeating 7 amino acid sequence where there's variation at places other than the first and the 4th amino acid, and then those are the nonpolar ones. And so we've given sort of each position in this repeating pattern a letter, A, B, C, D, E, F, G, and the A and the D positions are generally nonpolar. We also talked about how exposing nonpolar amino acids to water or nonpolar side chains to water is usually energetically unfavorable, in the same way that oil and water don't mix. And the reason for this is when you put nonpolar functional groups in water, they take up space that water molecules would normally occupy, and the water molecules in the immediate vicinity of the nonpolar side chains really have to slow down in order to hydrogen bond with each other. They can't interact with the nonpolar side chain, and so you get this ice-like layer of water molecules in the immediate vicinity of the nonpolar side chain. And if you rearrange the protein's conformation such that the nonpolar amino acids are not exposed to solvent, but rather are inside the protein, sequestered from solvent, then you liberate all of these water molecules that used to be immobilized around the nonpolar side chains. They get to have a lot more entropy, and therefore that process is thermodynamically favorable, because any process in nature is spontaneous if it has favorable enthalpy or entropy. And we could talk a lot more about this, but that'll be enough for us at this point. So our task at that point was to ask, are there particular structures that we could adopt that might place these A and D position nonpolar side chains in the interior away from solvent? And so we decided to look at the alpha helix, and here is a simplified diagram of the alpha helix. You're only getting backbone atoms. Of course, these dihedral angles, the phi and psi angles, are around minus 57 and minus 47. And that repeating pattern does a couple of things. First, it places a hydrogen bond between the carbonyl oxygen of the I position residue with the amide proton of the I plus 4 position residue. This is an I to I plus 4 hydrogen bond. And overall, if you count how many amino acids there are per turn of an alpha helix, you come up with the number 3.6, which is somewhat close to half. 3.6 amino acids is somewhat close to halfway through our 7 amino acid sequence. So if we tighten up that alpha helix just a little bit to 3.5, then every 2 turns of a helix, we come back to the beginning of our 7 residue sequence. So 1, 2, 3, 4, 5, 6, 7. That's 2 turns. Now we're back in the same place. 1, 2, 3, 4, 5, 6, 7. That's 2 turns. What this will do is it will tend to align the A and the D amino acids all along the same face of an alpha helix. So let me show you what that looks like. Here's the same sequence that we have above with the A and D positions highlighted. And this is looking down the alpha helix from the N terminus. And we're basically starting with the first A position residue right here. So this is that methionine residue, and we go from A to B, then C, then D. So the D and the A positions are separated by 4 amino acids. So that the D lines up on the same face of the helix as the A position. And then as you continue on from D to E, then E to F, and then F to G, and then we would be back to A for the next, starting with the next 7 residue sequence. We'll call that 7 residue repeating unit a heptad. And so what you're seeing is the first, what we've highlighted here below is the first heptad of this particular sequence. But the effect of this 3.5 turns, residues per turn, is to place A and D close to each other. And then if you continue down the sequence, this is what's called a helical wheel diagram. And it's the same thing that we've done here. The amino acids are listed in order from N to C, and you read the sequence like this: A, B, C, D, E, F, G, and then repeat. So the sequence would be M, K, Q, L, E, D, K, and so on. And that's so we've just superimposed this sequence on this wheel diagram, and what it's intended to show you is that at the A and D positions, you have nonpolar amino acids for the most part. So here is a stripe of 4 nonpolar leucine side chains. All of these look like this. And then at the other A position, at the A position rather than D, you have other nonpolar amino acids. There is a polar one in there. It has a specific role, but we're going to basically ignore it. Methionine looks like that. Valine looks like this. So you've got nonpolar amino acids here, and then if you look at the other positions, you should be seeing some different kinds of polar amino acids. So let's label in blue the cationic ones. You've got positive charges here and here. And then in, let's say, I don't know, red, we'll label the negatively charged ones, except that red looks a lot like what we did for the nonpolar ones. Oh well. Okay, so you can see that the other positions, G and E and B and C and F, all tend to have polar or charged residues. And there's more to be learned there. For example, you have 2 oppositely charged residues that are 1 turn apart here. Those are probably engaged in a salt bridge. But that's the general gist, is that if we fold this sequence into an alpha helix, we get, man, everything's going to just look the same over yellow, isn't it? Well, we'll just use gray. Well, this is terrible. I'm sorry. We'll circle the ones that are nonpolar in orange. That's a little bit better. Okay, so the general gist is you have a shape now that is greasy and nonpolar on this side, but is polar on this side. And this arrangement of amino acids is something that we call globally amphipathic. Amphi means like 2 or both characteristics, like amphibian. Amphipathic means it likes both. You have a part of the molecule, one face of the molecule that likes nonpolar environments, and then you have another face that likes polar environments. And so what will happen is that 2 or more of these helices will pack against each other such that the nonpolar surface area is sequestered from solvent. So here is an image of, I've taken actually this single helix from this assembly, which you can get from a crystal structure of a peptide derived from a yeast transcription factor. And this is what it looks like. You have two alpha helices that pack against each other and twist around each other. And this type of structure is called a coiled coil. It's a coil of helices. We sometimes call it a helix bundle. And if you look down the N-terminus, so this view here is sort of the Newman projection, as though we're looking down the, can't resist, can I? Looking down the N-terminus, and you see this view where you've got non-polar A and D position residues packing against each other. So here's a methionine from one helix, and here's the methionine from another. And then the next layer down, here's a leucine from one helix and a leucine from the other. And if we did a space-filling model of these side chains, you would see that they're basically touching each other. And that the fact that they are sequestered from water provides a lot of the driving force for formation of this structure. Okay, questions in general you want to ask about that or what's going on there? Yeah?
So if you put an amino or like a peptide sequence in like oil or something, would it just completely unravel?
If you put a protein like this in oil, would it completely unravel? Yeah, its structure would be different because you no longer have that driving force. And that question is actually relevant to proteins that are embedded in membranes because proteins in membranes have significant portions that are inside the lipid bilayer, and so they have to be comfortable in a nonpolar environment, and that their sequences are different in order to accommodate that. Good, others, yeah?
So if they have parts that are polar and they're non-polar, how are they assembled? Like what environment is the whole thing together?
So if they have, can you say that again? I'm not sure I totally got it.
Some is hydrophobic and some hydrophilic. What environment is the whole thing assembled together?
In what environment does the whole thing assemble together? It's a good question. The peptide is synthesized in the ribosome. Peptides and proteins are synthesized in the ribosome. And each amino acid comes in attached to a tRNA, which is a nucleic acid and is full of lots of hydrophilic groups. So as the peptide or protein is being assembled, it's in an environment that's okay with both characteristics. There's a little tunnel that the elongating peptide or protein chain exits the ribosome. And it's thought that as it exits and gets exposed to the cytosol, it begins to fold in that context. But and sometimes for some proteins you need helpers to get to the proper fold. These are called chaperones, and there's a whole area of research that's related to helper proteins that allow the protein you're interested in to get the right shape. Others? So this is one of the simplest sequences to design. These are, if you make a sequence that has nonpolar residues with this spacing of sort of A and D positions, it most likely will fold into a helix bundle of some kind. So this is one of the rules that's really well understood for protein folding. And just to give you an idea for how well understood it is, I want to look just once again at the nonpolar D and A position amino acids. Notice that the D positions here are all leucine, whereas the A positions vary a lot, but they have a lot of valine. And we said that valine is a beta-branched amino acid, meaning that at the beta carbon there's a branch point. Whereas leucine is not beta-branched, at the beta carbon it's just a CH2 group. And that looks like it's significant in the structure of the protein. It's thought, so here we're looking at one of the D position leucines, and it's thought that if we were beta-branched there, if we had, say, another methyl group, that those methyl groups would really get in each other's way. However, if you look at, this is going to be tough to see, but there's a valine side chain right here underneath the methionine. And you can see that the beta branching in that valine side chain allows, there's plenty of space. At an A position, there's plenty of space for the beta branching. In fact, beta branching allows you to fill space in the core that you wouldn't otherwise be able to fill. And so one of the rules that people have learned is that if you put non-beta branched residues at D and beta branched residues at A, probably like valine or isoleucine, probably you're going to form a dimer, because those particular shapes of side chains are best accommodated in a dimer. And years ago, somebody came along and said, hey, I wonder if we could change this sequence from a dimer to some other association state by playing around with these A and D position residues. And so there's a whole lot of ink that's been spilt on this subject, but now through really well understood rules, you can change the A and D positions and you can make a trimer or a tetramer, and then other variations have allowed you to make a pentamer or a heptamer. And again, this is there's this desire among chemists and biologists to be able to design amino acid sequences that do specific things, and this is one of the best understood areas in which you can do this. One, just zooming in briefly on the tetramer, you can see that in the tetramer sequence, all the other amino acids are the same. They've just played around with the A and D position residues. In this tetramer sequence, the A positions are non-beta branched, and the D positions are now beta branched instead of non-beta branched. So you have isoleucine here and leucine here. And that, I haven't shown you the core, the residues that are in the hydrophobic core to show you how they, how this particular shape, the shape of these side chains is best accommodated in a tetramer. But basically, you've got residue-based control over folding. A simple switch of where you put the beta branch residues changes the structure. Now, you shouldn't memorize that, but given data, could you work your way through it and try to understand? If I told you, hey, if you have beta branch residues at A, it forms a dimer, but if you have beta branch residues at D, it forms a tetramer. Design a tetramer sequence. Would you be able to know which amino acids to put at the various positions given that data? Okay. Questions? Yeah.
In these Helix bundles, is each strand identical, or can biochemists kind of engineer them to be different strands?
Cool. Yeah. So in Helix bundles, is it always that they're identical, or can you get different partners coming together? And the answer is yes. You can get hetero dimers, hetero tetramers, two different sequences coming together. One of the ways you could do that, we can see just by looking at this sequence. One of the ways that people have approached the problem of how do I make a heterodimer, that is, how do I have two different sequences that prefer each other? If you look at the sequence in this helical wheel diagram, these positions at E and G sort of flank the nonpolar core, and they tend to be charged. And so somebody had the idea, what if I took one of the two helices and made all of these residues at E and G positively charged, and then on the other partner I made them negatively charged. And that does create specificity actually for a heterodimer, where you have one that's positively charged, one that's negatively charged. So yeah, you can control that as well. Good. What else? Yeah.
I'm sure you've already talked about this, but with the heptad, when you're starting to label it, how do you know where to start with A, B, C? Like, where do you start?
Yeah, it's a good question. With the heptad, when you start to label it, how do you know where to begin? You look for the nonpolar residues, and there will be two possible options, right? I could have started here with A or here with D. So you just try them both out to see which one matches the pattern. If I had tried to start here with D, E, F, G, A, I would recognize, there's a mismatch there. I need my nonpolar amino acid at A, but it's at G. And so then you would try the other one.
You just find your first nonpolar one and start labeling and see if that works.
Yep. Yeah, you find your first nonpolar one, label it, and then try to find the pattern that best accommodates the rest of the nonpolar residues in your molecule. Okay, what else? Yeah.
Is it always in that arrangement, or does the order of the nonpolar change?
Is it always in this arrangement, or does the order of the nonpolar change? I think sometimes the first nonpolar amino acid is D. So, yeah, it's just the heptad defines not necessarily what the first one is, but rather it's the arrangement that fits all of the nonpolar residues best. Okay, others? Yeah.
Is there a difference between the amino acid sequence of a tetramer versus a heptamer?
Tetramer versus the heptamer, yeah. I think, in order to make this heptamer, they changed the amino acids in the nonpolar core to alanines instead of isoleucines and leucines. And they may have had to do some other things to the sequence, I'm not sure. But, and for the pentamer, they put tryptophan at the A and D positions, which is a larger aromatic nonpolar residue, and so you had to expand to be big enough to accommodate that. Yeah.
You have the beta branching on A versus D.
Yeah.
Different structures. Why do they take different structures than most of this?
Yeah, it's a little it's kind of unclear the way I've drawn it, and I do have some figures back in my PhD dissertation that would show this. So maybe I'll just. It's been like 20 years, not quite 20 years, it's been 15 or so, but let me try to illustrate it this way. Here is one helix and here is the other helix. In the dimer, I'm going to get this wrong. In the dimer, the A position nonpolar residues sort of splay outwards away from if this is the axis between the two helices. And so if you have beta branching, that tends to bring more nonpolar surface area back towards the middle. And so the A positions in a dimer can accommodate that kind of steric bulk, but the D positions in a dimer tend to point straight out into the sequence such that if you had beta branching there would be a steric clash. So the shape of the dimer can accommodate beta branching at A, but not at D. And it's exactly the opposite in the tetramer. And so that's why switching it works. All right, what else? Okay. By the way, coils show up a lot in viral fusion. So if you've ever learned about HIV and how that works, there's an important coil-coil trimer that turns into a hexamer that's involved in fusion of the HIV viral envelope membrane with your cell's membranes. And also, these kinds of coils are important in influenza, and I think coronaviruses as well. So it shows up a lot in places that you wouldn't necessarily expect. And understanding the rules has been really useful, and peptide and protein chemists have gotten a ton of mileage out of studying these things. In my lab, what we're working on is a lot of the rules are really well worked out for the A and D positions and also for the G and E positions. It's generally thought that you could do whatever you want on this solvent-exposed face, the B, C, and F positions. And we are learning that there are some long-range interactions between amino acids on the solvent-exposed face of the helix that can contribute substantially to how stable the coil-coil is, and we're still trying to learn how that works. So a lot of people are working on this, and it's a fun system to deal with. Another thing that I wanted to talk about that's a nice illustration of the relationship between sequence, structure, and function is the idea of antimicrobial peptides. So, if you've ever been stung by a bee, it hurts. And one of the reasons it hurts is because the bee injects, among other things, a peptide called melatonin. And that peptide is good at disrupting cell membranes, including red blood cells and other cells. And so there's damage done in the immediate vicinity of the sting. But a lot of organisms make antimicrobial peptides. These are short peptide sequences that can kill bacteria. The sequence we'll talk about today is called Magenin, and this is from frogs, and it's secreted from their skin, and it's named after the Hebrew word for shield. So why didn't the Marvel folks call the organization Magenin? Probably because it would be even more trouble to come up with an acronym that fit that than it was for shield. So, excuse me, here is the sequence. And here is an alpha helix. Now this doesn't look like the helical wheel diagram we saw above because this is not a coil-coil sequence. This is 3.6 residues per turn instead of 3.5. And what that means is that in 2 turns, you get a little bit farther than 7 amino acids down the line. And if you map this sequence out onto an alpha helix, the non-polar amino acids, phenylalanines here, isoleucines here, leucines and valines here, these all tend to be on one side of the helix, whereas the other amino acids tend to be on the other face. And most of these, a lot of these are positively charged amino acids. You've got lysines, histidine, and so on. Now, if you think about designing something that might infiltrate the lipid bilayer of a bacterium, what you would want, I mean, if you think about what the lipid bilayer is you have these polar head groups, and in bacteria, a lot of these are negatively charged. And then you have the non-polar tails that are in the interior. And so if you wanted to have something that could bind to the membrane and disrupt it, you would want a molecule that could interact with both the polar head groups that are negatively charged and the nonpolar interior. And this sequence actually does that. You have a nonpolar face that's globally amphipathic. This molecule is globally amphipathic, so you have the nonpolar face, and then you have the polar face back here. And it's thought that this peptide actually inserts into the membrane in a way that creates holes. So here is a view from a simulation that shows Magenin helices binding to the lipid bilayer and creating a hole. Here is a zoomed in view of what that might be like. You have the nonpolar face of the Magenin helix here on the left, which maybe I'll just color in gray, and that's interacting with the nonpolar tails of the lipid bilayer. And then you have the portion of Magenin that's polar and positively charged, such that it can oops, I need to get a little bit smaller font there, such that it can interact with the positively charged groups on the Magenin surface can interact with the negatively charged head groups. And for bacteria, once you open the membrane, everything inside sort of spews outwards uncontrolled and they die. So this is a mechanism that organisms use to protect themselves from bacteria. And Professor Savage's lab has a project where they've developed molecules that are not peptides, but they do this sort of thing. And it's thought that since most bacteria have membranes of this kind, that it would be difficult for bacteria to be resistant to this kind of treatment. And so ongoing development of molecules that target membranes and of bacteria is an important area of modern sort of antibiotic research. The trick is that you don't want the peptide you use to kill bacteria also to burst your red blood cells and make you blood clot and then die. So it's kind of tricky, actually. And Professor Savage's molecules are among the best in the world. They've got projects where they have coated their antimicrobial molecules on medical devices, and that prevents colonization of the medical device, catheter and whatnot with bacteria. I don't know if you've ever seen an endotracheal tube where they stick it down your windpipe and it fairly quickly gets coated with bacteria, and it's really nasty and can lead to other infections. But they don't pull it out and clean it out and put it back in, because that process would just shove the bacteria further down into your lungs. And so if you're on a ventilator for too long, you can start to get pneumonia and other things. But if you coat the inside of the endotracheal tube with antimicrobial compounds, the bacteria don't colonize it as much, and you can avoid infection. They've got a lot of interesting projects related to that, so really cool stuff. Okay, so I mean, there's so much more to learn in peptide and protein chemistry, but that's what we'll do for now. We're going to talk now about reactions, about how you might actually make these things in the lab. And there are two main ways that we do what is called peptide synthesis. And one is solution phase. That means we're doing the reaction in solution. And then the second is called solid phase, and that's a new innovation that we haven't talked about and that we'll learn. But both of these methods require protecting groups. So why would you need a protecting group? Well, you learned that earlier this semester that if you want to make an amide bond between an amine and a carboxylic acid, we use DCC, which is a coupling reagent, and usually we use some amount of non-nucleophilic base, in this case triethylamine, and that will make an amide bond. And we learned about the mechanism for that, and we're not going to review that here. But this is a great method for making amide bonds. The problem is, with amino acids, we'll just use glycine for fun. Your acid comes in the same molecule as an amino group. So suppose I wanted to make a dipeptide in which I had a glycine attached to an alanine. And suppose I just used DCC and triethylamine at this stage. Which carboxylic acid is going to get activated? Is one going to get activated in preference to the other? Probably not. And then which amino group is going to do the attacking? Probably either of them, right? So you're going to get a mixture of your desired dipeptide, where suppose we wanted the one where glycine was bonded to the amino group of alanine. So you're going to get this one, but, and this is the peptide GA, you're also going to get AG, and you're going to get AA, and you're going to get GG. And by the way, what's to prevent this from getting activated? And so then you're going to get GAA and GAG and so on. It's uncontrolled and it's a disaster. So we need a way to prevent an amino group and a carboxylic acid from reacting with each other in uncontrolled ways. So to do this in solution, we need a protecting group for carboxylic acids, and we're going to need a protecting group for amines. So for the carboxylic acid first, And remember, for protecting groups, and you've encountered these before, we used the TBDMS group for alcohols. We used acetal protecting groups either for alcohols or for ketones or aldehydes. But the goal for protecting groups is it goes on in high yield, it comes off in high yield. And if I'm going to have one for both carboxylic acids and amines, I'm going to want to be able to remove those protecting groups under different conditions, orthogonal conditions. So one that is often used for carboxylic acids is the benzyl ester. So here is the oxygen of the carboxylic acid, and then we have a CH2 group followed by a benzene ring. This is often abbreviated this way. BN stands for benzyl. That's different from phenyl. The benzyl group has a CH2 group in between the benzene ring and whatever you're attaching it to. The protection reaction is really easy. You take the carboxylate and you react it with benzyl bromide, which is just the alkyl halide version. We will call this BNBr for benzyl bromide, and via an SN2 reaction, you can very quickly put on that group, and oops, that's, there we go. And once it's on, once the carboxylic acid is protected as the ester, DCC won't modify that particular carboxylic acid. So that's a way you put a shield on a particular carboxylic acid so it's not reactive in a peptide synthesis reaction. And that putting on the benzyl bromide is a high yielding reaction. Taking it off involves a reaction called hydrogenolysis. It occurs under exactly the same conditions as hydrogenation of alkenes, but you take the benzyl ester, and then with hydrogen and palladium on carbon, you liberate the carboxylic acid, and the benzyl part pops off of the molecule as toluene. So basically, you're adding an equivalent of hydrogen across this bond. And the mechanism isn't important. Why it works has to do with the stability of a benzyl radical, but this works for bonds between benzyl carbons and oxygens. So that's a really high yielding reaction. It's also really efficient. You don't need to purify after you do it because you just make toluene as a byproduct, and that gets, and you can evaporate that off with the solvent. Okay, protecting groups for carboxylic acids. Any questions there? That's how we would prevent, if we'd had a protecting group up here, we put it on the alanine, then only the glycine would have been activated, and so we could control which carboxylic acid got modified. The second is for the amine, and there's a lot of different versions of this. Your text shows you two. I think we'll talk about one to begin with, and then we'll worry about that later. We want the requirements here. It's got to be stable to DCC. It also has to be stable to hydrogenolysis conditions, but it has to be easy to remove. And so the choice is tert-butyl oxycarbonyl or BOC. Some people say T-BOC, but that's really overachieving. Like organic chemists will say, come on, man, what other kind of BOC is there? So don't bother with T-BOC. That's a real try-hard thing to do, and you don't want to be try-hard. So the protection reaction looks something like this. And actually, you've seen this on, I think, exam 1, though you weren't really sure what it was and didn't actually need to worry about it. But this reagent is called BOC anhydride, or BOC2O. because this is the Bach group, but then you have this central section that looks like an anhydride. And so what happens is you can take an amino group and it will attack the carbonyl carbon to give you a tetrahedral intermediate. And then I'm cheating by not showing the proton transfer. Okay, fine. You would go here, and then a proton would need to remove, you would need to remove a proton from that nitrogen. Here's the rest of the molecule. When this electrons on that carbonyl oxygen kick down, the leaving group leaves and makes CO2, which bubbles off, and what you're left with is your amine that's now protected, and then a byproduct is tert butoxide, which picks up a proton from somewhere, becomes tert butanol, and you can evaporate that off or extract it away. And what you're the relevant thing here is this Bach protected nitrogen, which we can abbreviate like this. This functional group is called a carbamate, and it's a really useful functional group. And basically the idea is you have something attached to the oxygen that can be removed, and as soon as it gets removed, you have a CO2 that's just ready to come off. Let me show you how the deprotection reaction works for Bach. So the conditions for removing Bach are strong acid conditions, and the acid of choice is usually trifluoroacetic acid, or TFA. And what does this do? Well, let's see. All right, under strongly acidic conditions, here's the TFA acid. It's thought that the oxygen can leave from the tert butyl group to give you a tert butyl carbocation. And that tert butyl carbocation immediately gets picked up by, usually you have a little bit of water, TFA with like 5% water or something like that. So water will pick up that carbocation. Once you have the negative charge here, you're poised to spring out and get rid of CO2, and then the electrons here would go to nitrogen and pick up a proton from our acid. And then CO2 bubbles off, and you're left with your amino group. And I guess T-butanol as a byproduct. So Bach goes on easy, and it also comes off easy. But while Bach is there, this amine is not reactive, and because it's no longer an amine, it's more like an amide, right? Those lone pairs are tied up in resonance with the carbonyl group. All right, questions about these protecting groups? Yeah. All right, so with that in mind, let me show you now the good way to make our peptide. Let's see. Yes, the good way to make our peptide with glycine followed by alanine. All right, so first you would take glycine. Oops. And if you look at the sequence that we want want the carboxylic acid of glycine bonded to the amino group of alanine. This is our target peptide. So we're going to want to be able to activate the carboxylic acid of glycine, and we don't want this amino group to interfere. So we would use Bach anhydride to protect the amino group of glycine and make it unreactive. Similarly, for alanine, we want the amino group to be the nucleophile, and we don't want the carboxylic acid to interfere, so we would use benzyl bromide to, oops, I missed an alanine side chain. You could use benzyl bromide to protect the carboxylic acid. Then you add these two together with DCC and triethylamine. Now we only have one carboxylic acid and we only have one amino group. So the only thing that can happen is formation of the amide bond between the amino group of alanine and the carboxylic acid group of glycine. And then, because our target is this finished peptide, we have to do the deprotection reaction, which would be first, and you could do this in any order actually. You could use TFA and water to remove the Bach group, and then hydrogenolysis, hydrogen with palladium on carbon, to remove the benzyl ester, and that would give you your desired molecule. Okay. Now, what if I'm not interested in making dipeptides? What if I wanted at this stage to couple another amino acid here? What would I do? If I wanted to put another amino acid here on the N terminus? Could remove Bach. Yeah, is that?
I was going to say just remove one of the protected groups.
Yeah, remove one, not both. And then once you've removed this one, you can bring in another Bach-protected amino acid, activate the carboxylic acid, and put that on there. And this works really, really well, and people do this a lot, particularly in the Michaelis lab. But when you want to get out to peptides that are as long as what I told you about at the beginning of this class, like about 30 amino acids or longer, the individual yields of this reaction are generally not high enough that you can go stepwise and get out to 30 amino acids. And so we need another method, and that's solid phase, and we'll have to talk about that next time. We'll probably spend part of next time talking about it before we move on to the next chapter. All right, see you Wednesday.